today and tomorrow, I looked at the structure of the workshop and the different talks that were being given. I think a lot of them are focused a little bit more towards brain dynamics. Today's talk is not going to be about brain dynamics. Also, tomorrow's talk is going to be more about how to measure structure in the brain, how to measure structural connections in the brain. And the reason why this could be useful for brain dynamics is because that can allow you to develop these network models of brain function, right? So this is in some sense the first steps to identifying which areas connect to which areas anatomically, and that will allow you to then model each of those different regions as nodes and maybe with their unique properties, and then you can do dynamical models. But today is all about building these networks, right? Okay, so this first part is about how do you estimate structure in the brain, and as Dr. Stabra mentioned, a lot of this is thanks to Varsha Srinivasan, who was a PhD student in the ISC Mathematics Initiative PhD program. So for those of you who are interested, IMI is an interdisciplinary PhD program in ISC where people from different backgrounds can apply, and faculty propose various kinds of projects. So we proposed a neuroscience project, and Varsha fell right into the trap, and we got her for 5 years, 6 years to work with us. The other student who was involved in this work for today is Sawan, who was a student with Professor Partha Talukdar at the Computational Data Sciences Department. Also some of my collaborators and funding. So these are the two students who essentially made all of the work today happen, right? Okay. Also, since this is a small group, please feel free to stop and ask questions like anytime, right? So this is, consider this quite informal, so please feel free to stop and ask questions. Okay, so why do you want to study the brain's wiring diagram or the connectome? We've already said that it is going to help you develop dynamical models. But why is that useful? That's because the wiring is the basis for neural information processing, right? So the brain has, how many of you here know a rough estimate of the number of neurons in the brain? Anybody have a guess? So number of neurons? How many think it's 1 billion? 10 billion? 100 billion? It's about 100 billion, right? So it's about 100 billion, 86 billion neurons. So, but these neurons aren't working in isolation, right? They aren't working as independent units. They're talking to each other, and at the mesoscale, millions of neurons that form regions, in the brain are talking to each other through these extended axonal connections, right? These axonal fibers, and that forms the basis for neural information processing. So if they weren't talking to each other, a lot of the cognitive capacities that we know of wouldn't be able to happen. So it's very important to understand, how wiring works in the brain, because that ultimately produces your behavior, right? Because the language area, for example, has to connect to the muscles that control my jaw in order for me to be able to produce an articulate speech, right? So wiring is very essential for all kinds of behavior, and it's also important from another perspective, which is developing disease biomarkers. So there are certain connectomic disorders of the brain, and we will see that even other kinds of disorders that have not been generally thought of as connectomic have certain markers in the form of impaired connectivity. We will see that towards the end of this talk, right? And so these are all some of the reasons why we are interested in studying the brain's wiring, or the connectome, as it, if you will. Okay, so now the question is, if we want to study the wiring diagram of the brain, how do we study how do we estimate these brain connections in vivo, right? In vivo means in living tissue. Again, if any terms are not clear, please feel free to interrupt and ask, because this is meant for a sort of a mid-level audience, and so I haven't taken too much care to explain all the terms in detail. So please feel free to interrupt and ask if anything is not clear. Yeah. So how do you think we measure connections in animals, for example, connections in between brain regions of animals, where you don't have to do it in vivo, where you can sacrifice the animal at the end? Any thoughts about how those connections are measured, how different areas of the brain connect to each other? You don't have to do imaging in animals, right? Because you can sacrifice the animal at the end of the experiment. So yeah.
close to.
Exactly. So you inject some kind of tracer like a dye, virus, whatever it is, in one part of the brain, and then you see how that gets transported to other parts of the brain through these connections. And once it is transported there, you sacrifice the animal, you section the brain, and you can reconstruct how the tracer transported itself from one region to another, right? Now, unfortunately, you can't do that in living humans, right? So therefore, you have to come up with a method that allows you to get a good estimate of the connectivity in vivo, right? So how do we do that? So I'll give you a very high level bird's eye view in this slide and go into a little bit more detail in subsequent slides. So you have the human lying inside a regular MRI scanner. It looks exactly like the MRI you've seen in hospitals. The only difference is there's a slightly specialized sequence that's being used for imaging their brain. And this sequence, instead of measuring brain structure, right? So the normal MRI that you take is a static snapshot of the brain structure. But instead of doing that, this sequence measures the diffusion of water within the brain, right? That's why it's called diffusion MRI, right? And exactly what these little ellipses are, ellipsoids are, I'll tell you in a moment. Now, once you've done this diffusion MRI measurement, you go in, you apply what are called computational tractography algorithms. So this is where a lot of the math and the computing comes in, because this is where your estimation engine kicks in, right? Based on this diffusion, you have to now guess where these streamlines or axon fibers or fiber bundles are in the brain. And having made that guess, you can construct fairly elaborate diagrams like this connectome diagram, right? So this is in a nutshell, the principle of how diffusion MRI works. And I will go into each of the steps in a little bit more detail. Honestly, asking a neuroscientist to explain MR physics is like asking a software engineer to explain how a transistor works, right? So we know how to use it. We're not always aware of the principles behind how it works, but I'll give you my best understanding of how the physics behind MRI works. But it's actually fairly simple. The diffusion MRI is actually relatively, compared to some of the other kinds of MRI, it's a relatively simple idea. So the fundamental quantity that MRI measures is the spin, right? Is the alignment of the spins of the protons in the water molecules, right? That's the key quantity that MR imaging measures. So imagine that this is some chunk of tissue in the brain, and these spins are all sort of aligned in a certain way because you impose an external magnetic field, right, on this piece of tissue. They are all aligned in a particular way, right? This is T1 means at time T1, right, at a particular time. Now after some time, you impose what's called a gradient, right? So the magnetic field now goes from sort of a weak value to a strong value in a certain direction, right? So this is, remember, it's a chunk of brain tissue. Now you have the magnetic field going in a certain, growing in a certain way, right? Now what happens to these spins is that they all dephase, right? So they change their alignment a little bit, and that dephasing, because this end experience the weakest part of the magnetic field and this end experience the strongest part of the magnetic field, there's a sort of a monotonic level of dephasing across these spins in this direction, because this is the direction in which you impose the gradient. Is this clear? In this direction you are not imposing any gradient, so all the spins here are homogeneous. Does it make sense so far? Okay, so now we have done this. Now comes the trick, right? So you got your dephased spins in this piece of tissue, and then you apply a rephasing pulse, right? Essentially a rephasing gradient, and that gradient operates in the opposite direction, right? So now the strongest magnetic field is in this, at this location, and the weakest one is at that location, right? Everybody understands what a gradient is, right? Because it's a math group, I assume everybody knows what a gradient is, right? A gradient is anything that changes, right, in space or time and so on, right? So we are applying a smooth you're applying a sort of constant gradient here, going from a small value to a large value, right? So now what should this do? It should just reverse everything, right? You go back to how you were in the beginning of the experiment. So at T1, whatever the configuration was, all the spins should go back to that same alignment, right? That same phasing, right? Everything should now be back to the way it was at T1, right? Now this is if the protons are all static. In that mass of tissue that you're imaging, if the protons are not diffusing, if they're not moving around, if they're all completely static, this is what would happen. But what would happen if the protons started moving or diffusing, right? If the water molecules started diffusing, then essentially what happens is, between the time you applied the initial gradient and the time you applied this rephasing gradient, these water molecules, as indicated by the colors, have all moved off in different directions, right? So some have moved to this side, some have moved leftward, some have moved rightward, and so on. So now when you apply the rephasing pulse, they'll no longer be perfectly aligned like this, right? So you knock them off to one side. If they were in the same position, if you hit them with the same sort of rephasing pulse, they're going to come back to the upright position. But if they moved off, they're going to be hit with a different kind of a magnetic field, right? So they won't come back to the exact same position. Now what diffusion MRI measures is in some sense the resultant vector, the net vector, the mean vector, which is a measure of the alignment of these spins. So if nothing were moving, this final aligned spins would have a strong signal, right? Because the resultant vector is everything is pointing in the same direction. But because these things moved around, right, and the final rephasing does not happen to perfectly align them at the end, the magnitude of this vector is going to be smaller than this vector. That means you're going to see signal loss here, right? There's going to be complete rephasing and therefore signal loss. And so the extent of the signal loss defines the extent of diffusion. So the molecules are moving around a little bit, you will only see a little bit of signal loss. If they're moving around a lot, then you will see a lot of signal loss. Is this idea clear? So the diffusion MRI in a particular direction at a given time can tell you how much diffusion is happening. So it's only measuring a scalar signal, remember, at each voxel in the brain, at each point in the brain, it's only measuring a scalar signal. But then, if you really want to construct three-dimensional trajectories of fibers, how do you do that? It's very simple. You apply the gradient in many different directions, right? That's all it is. So the basic idea is that you apply this gradient not just in one direction, but between, depending on, how patient your participants are, between 30 and 200 directions, right? You can go up to a very large number. You're basically sampling on the surface of a sphere. I hope that idea is clear, right? So basically these are all unit vectors on the surface of a sphere, and so you can sample as densely or as coarsely as you want. So you can get this diffusion apply this diffusion gradient in any number of directions, and then that will allow you to estimate the diffusion along each of those directions, right? Now once you've done that estimate, you can construct what's called this diffusion ellipsoid. So if I take any point in space, I can, to a rough approximation, tell you the extent of diffusion along the three cardinal axes, but also along all the different directions that you sample, and then you sort of fit an ellipsoid around it. This is essentially the diffusion ellipsoid, right? This is there's a little bit more math behind this. You can actually model this as a tensor. I'm not going into that math in the interest of time, but the idea is fairly simple. So if you have white matter, so what's the difference between gray and white matter? Does everybody know what is gray matter, what is white matter in the brain? What is gray matter? Sorry? The cell bodies, right? Largely the cell bodies and also the dendrites, right? So in the cortex is the thin sheet of tissue that surrounds the brain. What about white matter? What is white matter? The myelin sheets, right? So what surrounds the axons, right? So in the white matter, because it's of its tubular structure, right, and because that myelin sheet that surrounds the axons is hydrophobic, water diffuses preferentially in certain directions. And so if you go in and measure diffusion within the white matter, you will actually see a preferred direction of diffusion. That's what's shown here, right? That's the ellipsoid. That's the major direction, major axis of this ellipsoid, right? Now then, once you obtain this diffusion ellipsoid at various voxels in the brain, because remember you're making this measurement across many directions in each voxel, but also across all the different voxels in the brain. So voxel is basically like a three-dimensional pixel, right? Across many different spatial locations in the brain. You will have a fairly elaborate map. I'll show you this in 3D in a moment, because what I showed you before was the correct 3D picture. And so based on the directions of these diffusion ellipsoids, you can trace a path. So your computational algorithm can go in and it can say, depending on the level of anisotropy. What is anisotropy? Isotropic means diffusion is same, right, in all directions. Anisotropic means there's a preferred direction. So depending on the level of anisotropy and the direction of this diffusion, you can actually trace this path and say, to my best estimate, there's an axon here, right? There's an axon bundle here, right? And then once you apply these computational algorithms, essentially what you get are pretty pictures like that, where if you start at a particular location and there's a fair amount of engineering and assumptions underlying this, just one moment, based on which you can reconstruct the path of this axon, path of these axon bundles. Yeah.
For each voxel, you had one order parameter kind of value, right?
That's right. Yeah, not one. It's a tensor. So every voxel, you have diffusion along. You may think of it as whatever n number of directions, right? That is the main thing. Does that make sense?
And depending on the magnitude in each direction, you build an ellipsoid.
Exactly. That's what this is. Correct. So we basically make an approximation. There are certain approximations involved in terms of how that tensor is built and so on. But once you build that tensor, you know the major axis of diffusion, right? Once you know the major axis of diffusion, your tractography algorithm exploits that to decide which way the streamline should go. This is the streamline. This curve here, right, is the streamline, and this is inferred, right? This is computationally inferred. There is no direct evidence that this exists in the brain, but there are studies. Obviously, we wouldn't be doing diffusion MRI if there were no proof for it. Okay, so actually that's one question. So let's say I build a very elaborate computational algorithm. How do I know whether it works or not? I may not know for a living individual, but is there anything I can do that to validate the algorithm in a non-living individual? Any thoughts what people might do?
We can do this for the animals which we can sacrifice while they're alive and then chop it.
Good. Yeah, you could do it with animals. Any other thoughts? You can actually do this in cadavers, right? You can actually do ex vivo imaging as well, and you can actually do this imaging and then sacrifice, then section the brain and then reconstruct some of this tissue. So that can be done, and that type of ex vivo imaging is also fairly.
Diffusion, so we operate in a similar way to.
No, that's a great question. So there are certain time windows within which you have to do it. So yeah. So that's a great question. So these are fairly well validated methods, right, that you can use for telling whether your computational algorithm works or not. But unfortunately for us, you can't do this in a living person, right? So for a given living person, you have to take your best guess using this computational algorithm. So that's what the rest of this talk is going to be focused on. How do you get the best connectome for somebody whose MRI signal you're measuring here and now, right? Actually, there's one more, and I sort of made a slightly different color on this slide because it doesn't directly relate to this talk. But you will also see a couple of other measures that people talk about, not just the structural networks. You'll also see a measure called fractional anisotropy, right, that people measure with diffusion MRI. And that is, if you fit this ellipsoid and you measure the, the lengths of these major axes, right? So the major axis, the minor axis, and so on. There's lambda 1, lambda 2, lambda 3. They're essentially the three principal eigenvalues. You can build a metric that captures the amount of disparity in these eigenvalues, and that's the fractional anisotropy. So if lambda 1 is very large compared to lambda 2 and lambda 3, that means there's a lot of directed diffusion happening, right? And if all the three are comparable, then anisotropy is less. So voxels where there's a lot of directed diffusion happening is where white matter structures are likely to be. And the reason why this metric is measured and favored is because many times this fractional anisotropy getting disrupted means the axon structure has broken down, right? And so that can be a marker of diseases and so on. And the other measure that you will see is mean diffusivity, which is the average of these 3 lambdas, right? This average is a measure of how much diffusion is happening in the brain. This can also be a useful marker for various kinds of neurological disorders. And then of course you can use all this information to construct the tractogram, right? So this is this is what we just saw a moment ago. So just to recap, so you put an individual inside an MRI scanner, you measure the 3-dimensional diffusion in every you measure the voxel, the diffusion along various directions in every voxel in a 3-dimensional map, and each of those ellipsoids is really the direction of diffusion along all those different voxels. You use tractography algorithms to infer how best you can fit streamlines to those ellipsoids, these diffusion ellipsoids, and then you come up with your best estimates of how these streamlines propagate across between different brain regions, right? So this streamline, for example, this tractography algorithm started in one part of the brain here, for example like the parietal cortex, and then sort of went down and decided that this streamline ended here, right? Similarly, this streamline started here somewhere near the occipital cortex and then went up towards the frontal cortex. So these are all things that you can estimate. It takes a lot of time to do this, but you can estimate and create these maps, right? These connectome diagrams. Okay. Any questions so far before I introduce the challenge? Yeah.
So because the diffusion gradient is, it is not abruptly ending. So how do you know this is the edge of an exon? Well, this is the boundaries.
No. So okay. So I'll be careful here. We don't know anything about axons, right? So right, or even bundles. See, the idea is the diffusion signal, the gradient is applied across the entire brain. It's not applied sort of bundle wise. It's applied across the entire brain, and in every 3-dimensional location, every point in space, you estimate this diffusion ellipsoid. Once you've done that, the computational tractography algorithm is what kicks in and decides where the fibers are and how they are going, and typically the start and end, and that's only associated with the tractography algorithm. The diffusion MRI doesn't know anything about this. The tractography algorithm decides the start and end based on the gray matter white matter interface. So that's the junction between the gray matter and the white matter where the axon bundles either go in or go out, right? So that's and that is not only to the tractography algorithm. Diffusion MRI sequence itself is agnostic to that. Right?
So tractography algorithm do have this direction. So going from this point to this to this.
That's right. So the diffusion MRI, that signal has a gradient. That has by direction, are you talking about A to B versus B to A? That we can't know. That information there. Okay, so when I said direction, I'm using it a little bit loosely, right? I don't mean direction as in a directed vector, because you can't ever tell whether the diffusion is happening, because the ellipsoid you saw was symmetric, right? So just go back here. So based on this, I can't tell whether the axon is running from region A to B or from B to A. All I can say is that there's a connection between A and B, right? That's all I can say, at least with this method, right? And you need the functional methods, etc., to get more detailed about the directionality of information.
What's the size of a voxel?
So that varies. So it depends on the strength of your magnetic field gradients that you can apply. So the ones that we use are typically about 1 millimeter on each side, but you can really bring that down, right? So if you know how to do MR physics well, which I don't, but we do know people who know how to do MR physics well, so we can actually bring that down to about 500 microns. So you can actually get very, very fine grained resolution within the brain. What's the big challenge here? Because it looks like, okay, we can estimate all of these connectomes, but the big challenge here is that because this entire tractography is an inference engine, one single MRI can produce many different connectomes, right? It looks like I pulled the rug from under your feet, right? So because if I have a single MRI, diffusion MRI scan, and this is actually a published image from a paper from my collaborator Franco Pestelli, a single diffusion MRI scan can give you both the connectome on the left and the connectome on the right. Now, looking at these two connectomes, can you take a wild guess as to what's the parameter that's different between these two connectomes, or an educated guess? Why does one look a lot smoother than the other? So, this is based on an algorithm called deterministic tractography, and this is based on something called probabilistic tractography. So, the essential difference is that in deterministic tractography, you follow the streamlines only along the longest axis of the diffusion ellipsoid, right? So, it gives you very clean, smooth streamlines. In probabilistic tractography, you also occasionally allow streamlines to deviate from that major axis, right? So, that's all. That's the only thing that's different between these two, and that can give you major difference in the streamlines that you reconstruct, and that again, it feels like now what do we do, right? Which is closer to the ground truth, because we don't have ground truth in this individual. Yeah.
These are two different connectors, or are they the same?
Is the data underlying
The underlying data is exactly the same. It is just the parameters, the tractography algorithm that are different. So, I did the diffusion MRI, right? Now I have diffusion along whatever 200 directions in each voxel. Now with that, I have to make an inference of what the connectivity is. Now how do I do that? That's the question, right? Does this make sense? And so if I use different parameters, I can get very different connectomes, right? That is the problem. So how do I know which is closer to the ground truth? Because we don't have, ground truth in terms of actual connectivity. Yes.
The second figure, you see more branched structures, right?
For exactly that reason, because you aren't constrained to follow the major diffusion direction, so occasionally you are allowed to veer off.
So does that say something about the direction?
About what?
Would it say something about the direction?
No, but based on the physics, you cannot in principle know.
Is it the case that near the source and target it tends to branch out more?
Again, I'm not exactly sure what you mean by direction, because that's a little bit of an overloaded word, right? So by directionality, if you mean from A to B versus B to A, that you simply cannot know, because the physics will not, at least whatever we have now, does not allow you to distinguish A to B versus B to A. That you can only know A and B are connected. You can't know the direction of information flow. For that, you actually need functional measures, right? So
was asking is this maybe there are two spots A and B,
yeah.
and near these two nodes they're supposed to branch out.
I mean, yeah,
And
asking, is
between
that the case?
it's more bundled.
Yeah, that happens, right? So, but you don't measure anything in the gray matter. This is all in the white matter only. So so what's another way of saying this? So you start from region A and you go to region B. You can either go to region A from region A to region B through some very well defined smooth path, or you can actually have a very branchy path going, right? So where you not only go from region A to region B, but also shoot off streamers region C, D, and so on. That's what's happening here, right? So essentially what we are saying is, if you're driving on a highway and you're constrained to follow like the fastest car, then you'll end up with a streamline like this, because you're basically always going in one smooth direction. But if you can occasionally follow a slower car, etc., then you can take branches and deviate. It's a property of the tractor. I mean, if you're interested, I'm happy to discuss with you in a little bit more detail, right? But again, you cannot infer directionality. Diffusion MRI does not allow you to tell whether information is going from A to B or B to A, right?
General question about this. So would this depend on like the activity state in the brain? Like certain ingredients you could make them more activated because it's stabilized, maybe more. I don't know. Just curious.
So that's a good question. As far as we know, there is no difference in terms of the underlying structure at the scale of activity, time scale of activity. So the structure doesn't. So it changes over the course of days, but over the time scale of, for example, activity induced by, for example, a task, right? If I'm asking you to pay attention versus rest inside the scanner, that's minutes. And so at that scale, to our knowledge, the structure doesn't change.
But I'm thinking in terms of like the activity, the diffusion and so on, might that be?
It's not sensitive enough to that level of activity change. Yes, this is all changes that can be measured over days. You can measure, like for example, if you're learning and so on, the microstructure can change, but not at the level of minutes. Okay, so how do we solve this problem, right? That's essentially the rest of this talk. And the answer turns out to be, I mean, this is again, our best guess as to what can be done, but there are probably other more, better ways to do it. The answer turns out to be connectome evaluation. So you look at the two connectomes and you make some guesses as to what might be closer to the ground truth. Now, how do we make these guesses? So for this, I'm going to give you a very different example, right? Just as a conceptual illustration. So imagine, any of you know what this figure is incidentally?
This was in the cover of the book Siddhartha showed yesterday.
Okay.
It's an optical illusion.
Correct. So yeah, so it's like a triangle that you can sort of perceive, whose edges you perceive due to Gestalt, right? So you can actually, your brain connects the edges and so on. So imagine this is a scan, right? It's some incomplete scan of some underlying, true connectivity. So three different connectomes, three different pictures are consistent with the information that you got from this scan, right? Because this is an incomplete, this is with some edges taken off either this picture or this picture or this picture. They're all, equally valid, connectomes that could have given rise to this scan, right? So these are three potential ground truths, and this is a scan you have. Your goal is to go from here to here. Now it looks like that's an intractable challenge, right? What is the solution? The solution, it turns out, is fairly straightforward, at least in the way that we implement it. We take a second scan, right? In the second scan, we are going to make an important assumption. We are going to make an assumption that these two scans are not perfectly correlated, and the aspects that are perfectly correlated are signal, and the aspects that are not correlated are noise, right? That's the assumption. Noise is uncorrelated across the two scans. Signal is going to be strongly correlated across the scans. Now let us say you take one more scan. Now you can constrain, right, which is the most likely of these three pictures to have given you both of these scans, and that's likely to be this one, right? The other two are not potential candidates that could have given you both of these scans. Is this idea clear? At a very high level, right? Now once you jump into the math, this is essentially it. You measure the diffusion signal, you use your connectome algorithm to estimate the connectome, you predict the diffusion signal itself, right, from the underlying connectome, and there are ways to do it. You have the connectome, you can go back to the diffusion signal, and then you measure the difference between the measured and the predicted connectome. So this algorithm that Franco and colleagues developed is called linear fascicle evaluation, and what it allows you to do is it allows you to estimate weights for each streamline. Each of these connections that your estimation algorithm has inferred between two different brain areas. So you want to estimate weights that minimize the error between the measured signal for one scan and the predicted signal from another independent scan, right? So essentially you've taken a scan, you sort of created a connectome and you're predicting the signal, and you're going to look at how well it matches the signal that you obtain from another independent scan. Does this make sense? So because there are a lot of weights that are completely, streamlines that are useless for predicting, right? Those will all be assigned 0 weights. So you could have traced out millions of streamlines that are completely useless for predicting the signal in the other scan. Maybe they were only just driven by noise, and so those would all get assigned 0 weights, and only the ones that are actually predicting signal across both the scans would be retained in the final connectome. Some people also call this connectome pruning, but we call it connectome evaluation, right? Is this rough idea clear? Yeah.
How will another independent scan have the same signatures of the measured diffusion?
The same individual. Yes. Sorry, I wasn't. Yeah, sorry, I wasn't clear on that. So it's from the same individual, not from a different individual. From the same individual. Again, we are hoping that across the two scans, signal will be correlated, noise will be uncorrelated, right? That's the idea. Yeah.
Like every time you do cryptography, you get the same connectome.
So that's the idea. You can derive any number of connectomes, but once you do connectome evaluation, the fibers that you're left with at the end should be same or similar across all the different tractographies that you've done.
Choose that connectome which has the same, in which the predicted diffusion and the measure are approximately same.
Are approximately similar, but we phrase the problem a little differently. You overestimate the number of fibers, you go with the most liberal method, trim it down to the point where this match happens.
Like in spectrography, you have to input the number of fibers you want.
Correct.
So you get that to maximum and then prune.
And then prune it down. Exactly. That's why others prefer to call it connectome pruning. So, yeah. Is this rough idea clear? You're basically saying I want to paint a very complex portrait, then I want to trim it down until it actually looks like the ground truth, right? Okay, so this allows you to have fairly good estimate. Why do we think this works? I'll show you this in the last few slides. Why do we think this is useful? Yeah.
The previous slide.
Yeah.
How do we choose the neurons that we don't need? Like how do we trim it down?
So what we actually do here when estimating the whole brain connectome is we look for connections from any part of the brain to any part of the brain. Large number of connections, right? So it's like letting a garden grow wild, right? You got lots of connections, lots of plants in the garden. Then you decide which matches the ground truth. Which of them is, if you try to reconstruct the diffusion signal from this connectome, which matches the ground truth of a different scan that you got from the same individual, right? And so you prune the weeds away, you prune out all the unwanted connections, right? So that's the important point, right? So that's a good question. The important point is that even if you measure the signal again, you expect that the components that are actually signal will be correlated across the two scans. That will be the same, repeatable. Anything that is noise, right, will be uncorrelated. If noise ends up being correlated across the two scans, you have a problem. You can't do too much about it, right? So if you get the same repeatable noise over and over again, at least this method can't take care of it, right? Does this make sense?
Yes.
So these streamline waves are something with the number of bundles.
Number of bundle. So Franco sort of loosely calls this the volume of the streamline, right? But you can think of it as sort of in a very abstract sense, it is essentially what I've put there. It's the evidence that streamline carries in terms of its value for estimating the ground truth diffusion MRI signal. It's sort of the strength of evidence for that streamline in the data. You can loosely think of it as volume, but it could be a bit misleading, right? You can think of it as the sort of the volume of that stream, but that's a bit misleading. Yeah.
That's assuming that the many connectomes that we got from MRI, say using deterministic and probabilistic tractographies, that they differ primarily due to noise rather than different methodologies, like different approximations being taken at the two approaches.
That's while that is accurate, the reason why they would produce different connectomes is largely because of them fitting noise. So you want them to fit signal. So while what you're saying is correct, empirically it turns out that mostly it's because they're trying to fit all kinds of irrelevant small fluctuations in the data, right? Loosely it's like trying to overfit a curve to like noisy data, right? So you just want to sort of regularize that, and that's the essential idea here.
Deterministic tractography will, without even this method, be similar across multiple scans because that is already over smoothing the data.
But that may be underfitting, right? So then you have, if you correct for it, it may actually give you better solutions. Yeah, that's a good question. Any other questions? Good. So the rest of the talk is just advertisement, right? So basically, what is the advertisement for what we did? What is this linear fascicle evaluation algorithm was developed right around the time I was finishing my postdoc, and so we were quite excited that we could apply this in our data and so on and so forth. And so the advantages for it are it prunes fibers by evaluating fit to the underlying diffusion data, and it's very memory efficient and so on, because these are large streamlines, right? So Franco and his collaborators have done a very good job of making this very memory efficient. But what are the disadvantages? So when I started my lab in Center for Neuroscience, we had like basically a couple of computers to work with, desktop computers, and pruning a 1 million fiber connectome is generally a regular, that's typically what the number of fibers that you generate, right, for these types of applications. So that took an entire day for one participant, and we were like, they're never going to be able to use this effectively, right? Especially because we have these large databases that we now have access to. Now I think UK Biobank has like maybe 10,000, maybe an underestimate. I think they're probably 100,000 or something. There's a huge database of scans that are available. So we were like, this is not even going to work for small data sets that we run in the lab, because we change something a little bit, you have to wait for weeks and months before you get your pruned connectome again, right? So then what we did is, in collaboration with our colleagues at computational and data sciences, we developed this approach called regularized accelerated linear fascicle evaluation, or real life, and all it does is faster and more efficient pruning of the connectomes. So how is it faster and more efficient? I'll show you in just a moment. So it's a little bit of math here, so bear with me. So you can think of this process of reconstructing the diffusion MRI signal from the connectome as a matrix decomposition operation, right? So a non-negative matrix factorization operation. The basic idea here is, these are all this matrix here represents your different streamlines of fibers, right? I'm not going into details of the what each of these indices represent and so on. I'm just giving you a very high level overview here. But this is in essence, each of the fibers that you've estimated with the connectome, a matrix representation of that, right? And if you, just in case you're interested, NV is the number of voxels, N theta is the number of directions along which you sample the diffusion signal, and NF is the number of fascicles, right, the number of streamlines that you've estimated. And this vector here is what life is estimating. So this is a weight associated with each fascicle or each streamline, right? This is the unknown, right? This is what we want to estimate. This is the measured diffusion signal, right? This is what you measure. This is the number of voxels times the number of directions. I already told you that in each voxel you're measuring in many, many different directions. So this vector's dimensionality is NV and theta, right? So this is what your measurement, this is what your streamline algorithm estimated, and these are the weights that you want to infer using some minimization, optimization like this, right? You want this product to be as close to this measured signal as possible. Now it turns out that you don't have to do this on CPUs. GPUs, because some of these computations can be very easily parallelized. GPUs have thousands, now millions of threads running in parallel, right? So you can actually take these matrix vector multiplications and factor them out into many GPU cores, and it turns out that you can achieve lots of speed ups if you do this. And this is exactly what Sawan, the CDS PhD student, did in collaboration with Varsha. And what he was able to show is that this is the speed up, not in percentage, right, actual speed up factor, right, for various different data sets. So this is the Human Connectome Project data set. Some of you may have heard of HCP. This was a data set that was acquired in ISC, and this is a data set that Franco used as benchmark, acquired at Stanford in his paper. And what we find is that if you increase the size of the data set in terms of the number of fibers in million, you actually get more and more speed ups, 70x, 80x speed ups depending on the size of the data set. And the nice thing for us is that these speed ups scale up for larger connectome sizes. I mean, they do saturate, but it doesn't become slower as the connectome size becomes larger. So that's a good thing for us. So if you have large connectomes, you can actually prune them quite efficiently. Okay, so this was the first version. This was published in a conference, and then we also have a journal version of this now, where Varsha took this and advanced this even more. And so now we have speed ups, of the order of 160. Now we have, the latest versions are up to 200x speed up compared to the original CPU implementation. And this division is just a cute way of saying the CPU relative to the GPU. And this was covered on the cover of Nature Computational Science because of this seemed to be sort of a very interesting thing for the editor. And so normally the reason I put this cover image is only partially for bragging, right? Is one of the rare instances where normally you submit cover images to journals for consideration, and then they select from some of the pool of images. And then so in this case, Varsha was finishing her PhD, and I sort of begged her to submit a cover image, and she said, no, I'm too busy, and she left. But the editor themselves selected some stock image from the internet and made this into a cover image and published this paper. So it's I thought some of you may not understand the joke, but it's like a rare thing to happen, right? The editor pulling some stock image and deciding to make it the cover, but it was it was one of these rare sort of happy moments for this getting covered. Okay, so then why do we believe this is this is useful, and why do we think this is doing any better than the actual real life algorithm? Actually, one thing that I forgot to tell you, and we should probably scroll back, is you may notice that there's a difference between the objective functions of life and real life. So life, all it's doing is minimizing this error, right? So it's minimizing the error between the measured diffusion signal and the reconstructed diffusion signal, just this Bm minus Mw. Real life, in addition, has a penalty. There's a penalty term associated with having non-zero weights here. This is an L1 regularization penalty. Was there a question? No, okay. It's an L1 regularization penalty, which means that if you have too many non-zero weights here, you get penalized. So you essentially estimate sparse connectomes, right? So that's the idea. The idea is that you any weights that are close to 0 get driven to 0, because L1 effectively acts as an L0 regularizer, and so all these weights that are very close to 0 get driven to 0, and so then we estimate sparser connectomes as well, right? Now how do we estimate lambda? That's a different question for a different talk, but if we do this, what happens? So this graph is a density plot of a large number of streamlines estimated across participants, and on the x-axis you have what's called within participant variability across different scans, right? And on the y-axis you have a measure called between participant variability, that is between the connectomes estimated between two participants. What you want is a large between participant variability and a small within participant variability. So if you estimate the connectome over and over again and prune it with this method, you should have relatively minor variability within a participant and more variability between participants. With life, you see that that doesn't always happen, right? For a large number of streamlines that we estimate, they are sort of balanced. But with real life, this is the sparser and faster, of course, connectome estimation approach. Your within participant variability goes down significantly. Your between participant variability also goes down, but not as much, right? So this ratio of within to between, which we want to keep as low as possible, is better with real life than with life, right? So this is some evidence that this pruning technique is estimating more consistent connectomes more consistently, right, for the same participant. Then we also applied this method. Like I said, the rest of it is going to be just advertisement, right? So we also applied this method to just one moment. Applied this method to a tractography challenge, and in this challenge, essentially what you have here is a ground truth set of ground truth bundles. So these are simulated connectomes that the this ISMRM folks have given us, and in these simulated bundles, once you finish your estimation and pruning, you want to have as much overlap with the simulated bundles as possible without having too much overreach. Overreach means your the bundles that you estimate shouldn't fall outside the ground truth bundle. The overlap means you should have as much sort of overlap with the ground truth bundle as possible, right? So you can construct a graph like this, plotting overlap versus overreach. So if you have a very, very liberal connectome estimation method, it will have a very high overlap and a very high overreach, right? So you can estimate streamlines from every region to every other region, and then you will end up with a large overlap, but you also have a large overreach because you have estimated a lot of incorrect connectomes. Very conservative method would be at the other end, right? So there's some kind of Pareto front here, right? So these other dots here are other people's methods, and so what you would ideally like to have is large overlap with little overreach, right? So you would like to be somewhere here in the plot, and what we find is that these colored dots here are our methods. They are ahead of this front here, right? So they are more towards this upper left than any of these other competing methods. Okay, what was the question? Yes.
Does it depend on which brain region is the optimal method?
So that's a great question. So you can tune it in many different ways. So right now we've only fixed it to one value based on a validation set. So but you can tune it in many different ways. That's right. We haven't done that yet. So but you're welcome to try. The code is available. Yeah. And we also showed that we perform better than another competing state-of-the-art approach, which is called SIFT, in terms of this is just a quantification of some comparison here. Okay, so in the remaining 5 minutes that I have, I'll go over like a couple of very quick applications. Where can you use this? So one of the nice things about structural connectomes is that they're fairly stable over days, weeks, months, sometimes even years, right? So in that sense, you can actually use the connectome, structural connectome, as a basis for estimating inter-individual differences in cognitive traits. So that's exactly what Varsha did here. So what she did is, took vectorized features from this structural connectome, and you can convert this connectome into a matrix, right? So this part maybe you've all seen before, right? So where you have the one set of regions on the x-axis, another set of regions on the y-axis, and a value for I comma j on each entry of the matrix, which corresponds to the weight estimated by life or real life for the connections between those pair of regions, right? I and j. Now, for those of you who've been following along very carefully, why does this matrix look upper triangular? Why are there no entries in the lower half? Only those who've been following this very carefully can answer this. The connectivity matrix, why is it not? Something we said about directionality, right? It's symmetric, right? It's essentially symmetric, so there's no value to having information in the lower half, right? I mean, you could have put in information there, but there's no value to having it, because the way diffusion MRI estimates connections, I to j is the same as j to I. Based on the physics, you can't distinguish, right? So the connectivity strength is between two areas. Remember, it is not directed. So if somebody tells you, I measured with diffusion MRI this area connects to this area and so on, that's an incorrect way of expressing things. You can only tell that these two areas connect with each other, right? So based on this connectivity matrix, you can run what's called a regression model. So you take this connectome, weights as features, and then you can try to predict things like various kinds of cognitive scores, right? Language scores, reading scores, things like this. And what we found is there's a slightly interesting way of plotting things, which is based on the certain significance threshold for these predictions. You plot the average predictability, right? And generally, the predictability is higher with this pruned connectomes with real life as compared to the unpruned connectomes, which are the default algorithms connectomes. And I mean, Varsha showed this across many different tasks, and the ones that actually stood out in terms of their predictability of their scores were tasks of cognition like reading, and tasks involving naming of pictures and so on and so forth. And some of this is, yeah, good. That's a trait, right? So the Human Connectome Project database has lots of traits and lots of cognitive scores. They also have a loneliness score associated with how lonely somebody is feeling. So there's lots of different scores that are available in that database. But this, our ability to predict some of these is a function of how well we can also estimate the connections between two regions, right? There's a very, very strong fasciculus, a connection between different areas that process language, and that's why we believe we can, predict these traits very well, these scores very well, the reading, English, picture vocabulary, and so on and so forth.
So these values, they mean exactly what? So this bar plot.
Yeah, so I didn't go into this in much detail in the interest of time, but the basic idea here is if you merge the features from two different methods, one is the method that is using real life pruning and the one without pruning, when the algorithm makes its final prediction, which features does it favor? So, if it favored both features equally, you would lie exactly along this 50% line, that is this dash dash line, right. But if you favored real life features more than the other methods features, then you would be above this line, and so invariably for most of the scores you are above this line, suggesting that it prefers those features, probably because they express a more consistent relationship between the connectivity strength and behavior compared to the unpruned connectomes features. So, but yeah, I mean, I didn't go into that in the interest of time, but that's essentially what that is. So, you can do, I mean, anyway, so I'll just skip to the end. There's lots of interesting things that you can do, and those of you who are interested in this is a very, hot topic these days in terms of predicting brain age, in terms of dementia. How many of you here have heard of this brain age? Some of you, okay, fantastic. So, the basic idea is that chronological age is not a great predictor of, how somebody's brain ages. Using structure and actually now connectivity, you can make some predictions about how the brain ages, and if that predicted age is higher than the chronological age, then perhaps something is accelerating the aging of the brain, right. So, you may want to caution the individual to be more careful or whatever, right. So, at least that is a diagnostic marker, it looks like there is some value to it. And conversely, if your brain ages more slowly than chronological age, it's sort of looking younger than the person is in actual years, right. So, those are some interesting things that you can do with this.
Just from an experience, yeah. Do you have some idea of, from the structural data itself, what would it mean to have a younger brain and say 30 versus 45?
Correct. That's what. So, the younger brain, older brain, there is a certain normative age range within which this analysis should be done. So, I should say that carefully. It is much more relevant for as people age, right. So, past 45, past 50 and so on. So, that is when this analysis becomes really useful, and the idea there is if your brain has the characteristics of somebody who is 30 when you are actually 60, then it is possible that your brain is somehow healthier, right. That is the idea. That is the.
Is it based on level of neurodegeneration, or I don't?
That is the idea. The idea is normatively. Some parts of the brain would have, some parts of the gray matter would have shrunk, some parts of the white matter would have gotten, dissociated and so on. Just normal aging, right, and through normal aging. But does your brain show accelerated signs of those pathological signatures, or deteriorated signatures, or does it show less, slower, rate of decline of those signatures?
From the same individual.
So, you could do that. Those are called longitudinal studies. This is all cross sectional, where you are looking at a population, and you assume that everybody who is like around 70 is aging normatively, etc., and you compare the outlier individual to see if are they above, below, etc. So, I mean, yeah, so you get the broad idea, right. You can play this game in many different ways, and so what we did is we did the same thing, but using connectomes instead of using brain structure. And you can, what we showed, and these are some preliminary results, is that when we looked for the connectivity correlates of brain aging, we actually found a certain connectivity in the parietal cortex that was critical. But when we looked for connectivity correlates of cognitive decline, right. So, these participants whom we had tested had also performed various kinds of cognitive tasks, and looked at how steeply those scores were declining, and that predicting those cognitive scores seem to correlate with a different part of the brain. So, it appears to us at least that aging versus cognitive decline are driven by different subnetworks in the brain. Again, in the interest of time, I won't go into this in too much detail, but you're welcome to talk to me afterwards, and also, yeah.
What is it that normalization is necessary? Is the chronological age?
What is necessary?
X-axis. You see the chronological age is like a normalized values.
That's right. So, it's not necessary.
It's not necessary.
It's just for plotting purposes. That's correct. Yeah. So, these are essentially derived from factor analysis of these cognitive scores, just to keep them on the same scale, right. That is the only reason. Yeah.
So what does age mean in a practical sense if it's not if it's separate from cognitive skills? Is it likelihood of getting dementia, or what exactly does
That is exactly what we're trying to say here. That just because somebody is aging doesn't mean they are also likely to be declining cognitively. Those two things seem to be dissociated.
Oh, like brain aging?
Just normally things can go, like I mean, when they get develop wrinkles in their face, right? That doesn't mean there's a deterioration necessarily, functional deterioration. Right? So, okay, and then again you can, many of you may have heard of this ADNI database that has scans from Alzheimer's patients, and we also in this AAA paper which also showed that real life has this interesting application where if you actually use the real life weights compared to things like numbers of fibers in the unpruned connectome and so on, and actually do slightly better at predicting classifying Alzheimer's from age matched healthy controls. Right? Alzheimer's disease patients from age matched healthy controls. Okay, so I'll quickly summarize. I'm out of time. So the structural connectomes are the basis of behavior. So we've shown you some very high level views of how you can use diffusion MRI and tractography to estimate these connectomes, and the challenge is one of indeterminacy, right? Or it's basically an underdetermined system, and so therefore can we use connectome evaluation in some way to address this challenge? So we try to make the system a little bit more, add more constraints and see if it can do slightly better. And real life is our approach for actually making this fascicle evaluation faster and more efficient on GPUs, also more accurate, hopefully by adding that regularization constraint. And then you can use these types of pruned connectomes as a basis for cognitive function, either healthy cognitive function or pathological decline like in Alzheimer's disease and so on. Right, great. So that we're hoping to extend this in various different ways. So discussions with in terms of various collaborations for large scale data analysis, because that's what really this approach is useful for, analysis at scale. So thank you for your attention.